Hello, friends. Welcome. My name is Bonnie Schmidt, and I'm so glad that you're here. I am a Neurosciences Master Trainer, and NEAR stands for Neuroscience, Epigenetics, Adverse Childhood Experiences, and Resilience. And I'm so honored to be here today as your host for this presentation on epigenetics. And all of the information that you will see here today in the slides comes from the ACE interface program, which is the program that I received my training through. And there's other information that I've pulled from different resources about epigenetics that I will add to the information packet. All right, let's get started. I'm going to share my screen here with you. Here we are. So I've added my email here for you. If you have any questions about this presentation or any of the neurosciences information, please feel free to reach out, email me anytime. I'm happy to answer your questions. I also just want to say a quick thank you to Clallam Resilience Project and Kitsap Strong for the work that they are doing to build resilience in their communities. So I'm so happy to be here with all of you. All right, so epigenetics. The word epigenetics literally means on top of your genes. So epi, on top of, and genetics is your genes. So before we dive into the deeper study of epigenetics, we need to talk a little bit about DNA. So within most of the 50 trillion cells that are in the human body on average, there is 6 linear feet of DNA. And that means that within the cell nucleus, which is like the brain of the cell, that the DNA can be stretched out to be as tall as 6 feet. Oh, it's so amazing. So how in the world does all of that DNA fit into the cell nucleus, which is approximately 400,000 times smaller? Well, this happens in a couple different ways. And the first way is a beautiful double helix structure that most of us are familiar with, that we've come to know about DNA, right? Well, the second is a more complex packaging solution, which has the DNA wrapping around proteins in the nucleus that you can see here, which are called histones. So you can think of these like molecular spools, like a spool that you would wind thread around, right? And there are approximately 30 million of these histones within each nucleus, 30 million. So the combination of the histone plus your DNA gives us something called chromatin. So now in your DNA, there are genes, and these genes are the instructions for the cell and tell each cell what to do and what kind of cell to become. And in our body, we have lots of different kinds of cells, right? We have brain cells and skin cells and blood cells. And these genes become tightly packed in this chromatin structure. And so the cell can have a hard time reading them. So this is where epigenetics comes in. So epigenetic tags or markers come down and are laid on top of the genes, and they give instructions to the chromatin to either compact or decompact. So either to coil tightly or uncoil. I think of this kind of like a slinky. If you've played with one of those and you pull it out and then you let it go, and so it gets really small. So through this process of compacting and decompacting, right, coiling and uncoiling, certain genes are hidden while others then are revealed, right? So this sequence of readable genes in turn then gives information to the cell and instructions about what type of cell to become and its function. So what makes the cell look different, like the difference between how a neuron looks from a muscle cell or a skin cell, is these epigenetic markers. And each cell has the exact same DNA, right? Each cell has the exact same DNA, but the epigenetic markers causing the DNA to coil and uncoil or compact, decompact in different areas produce different types of cells. So epigenetic information first starts to get laid down during embryonic development. So when cells start to divide, they start talking to each other and they receive information from each other as more epigenetic markers are then laid down on the chromatin. So now we know through research that these marks can be influenced by the environment. So here's where this is relevant to adverse childhood experiences. New research is really suggesting that stressful experiences can influence these chemical attachments and influence the reading of our genetic code in ways that can have negative effects on the way our cells function. So this may be a really important reason why adverse childhood experiences are related to so many health and social problems. The hopeful part of this is that other types of positive life experience may affect and impact the reading of our genes that in turn may reverse the negative effects of childhood adversity or have positive effects on human development and well-being. So scientists have discovered that responses to stress may be passed from one generation to the next. In one study, scientists conditioned rats to be afraid of the smell of cherry blossoms. And when the cherry blossom odor came into the rats' environment, they would huddle together all stunned with fear until the odor was removed. Well, when these rats had pups who grew up without any exposure to cherry blossoms, this generation of rats was then separated from their parents and placed into a different environment. When the smell of cherry blossoms was put into their environment, the rats were super jumpy, they had heightened sensitivity, and were just agitated at the smell of the cherry blossoms. So why might it be a good thing for sensitivity or fear to be passed from one generation to another through epigenetics? Why might it be a good thing for the rats to feel fear, even though no one instructed them to fear cherry blossoms? And the answer is simple. It's for survival. Fear can protect a species from dangerous situations or toxins, and it may be adaptive for fear to be passed down from one generation to the next if it can prevent poisoning or help a person avoid a danger. So the field of epigenetics may help us understand how groups of people may be deeply affected by historical trauma, despite current efforts to care for one another well. And so how do we heal the wounds of our ancestors? And we may not fully know the answer to that question yet, but we do know that we are the ones responsible today because we are the adults and no one else is waiting in the wings. So let's think again about the positive side of experience being passed from one generation to the next, because wherever there is a history of trauma, there is always a history of resilience. And scientists are testing whether positive characteristics like nurturing behavior can be inherited from one's parents and grandparents. The field of epigenetics provides us with hope that our ways of relating to one another can create lasting positive changes in genetic expression, how creating self-healing communities now can lay an enduring path of generational healing and recovery. It's up to us, right? All right, so we are going to watch a short video that I just love that explains epigenetics in a very relatable way. So I'm going to pull that up for us now.
Writing's not that easy, but Grammarly can help. This sentence is grammatically correct.
The sweet smell of fruit doesn't normally send rats running, but when researchers paired the orange cherry almondy scent of the chemical acetophenone with a painful electric shock, lab rats quickly learned to fear it. Along the way, extra neurons sprouted in their noses and in the smell processing center of their brains, making them super sensitive to the scent. This result isn't shocking. What is surprising is that the rats' pups and their pups' pups were also startled by the smell of acetophenone and had the same extra neurons as their fathers, despite never having been introduced to either their dads or the fruity scent before. But how could the pups have inherited something that their fathers learned? Basic genetics tells us that only DNA gets passed along to offspring. Characteristics like memories, scars, or giant muscles can't get passed on since acquiring them doesn't alter the genetic code. But it turns out that instilling fear in the rats did trigger genetic changes, not in the DNA sequence itself, but instead in how that code was read and used in the rats' bodies. In every cell, biological machinery constantly translates DNA into the proteins needed to carry out vital processes. Chemical switches attached to the DNA turn genes on or off or up and down, telling the machinery which proteins to produce and in what quantities. These switches, called epigenetic tags, are why a kidney cell looks and acts differently than a skin or nerve cell, even though all three cells have identical DNA. But the switches in any one cell aren't set in stone. Teaching those rats to fear the fruity smell switched one of their smell sensing genes into overdrive. Researchers don't know all the places in the rats' bodies where this switch got flipped, but they know it happened in one key set of cells, the rat's sperm cells, which would one day pass along the tweaked genetic material, making the next generation of rats super sensitive to acetophenone. Rodents aren't the only creatures demonstrating this weird type of inheritance. In Epigenetic, Sweden, boys who suffered through tough winter famines went on to have super healthy sons, with extremely low rates of heart disease and diabetes. And their sons had the same excellent health, living an unbelievable 32 years longer on average than the grandsons of boys who hadn't gone hungry. To be clear, this does not mean that we should start starving our kids for the benefit of future generations. Scientists don't even know yet exactly which switches the Swedish famines flipped. While we have been able to connect specific epigenetic changes to health effects in mice, we're a long way off from being able to make those connections in humans. That may sound like a bummer, but it's mostly because we humans don't live in the well-controlled environment of a laboratory. And for that, we should be grateful.
So, the field of epigenetics really provides us with hope that our ways of relating to one another can create lasting positive changes in genetic expression. How creating self-healing communities now can really truly lay an enduring path of generational healing and recovery. So thank you so much for being here with me today. If you have any questions, please feel free to reach out to us at www.clallamresilienceproject.org, and we will be so excited to hear from you. Hope that this finds you well and that you're having a beautiful day. Bye.